to somebody who's very well known to us here at the Brain and Mind Centre, and that's Dr. Max Shine, who's a Robinson Fellow, early career researcher here at the BMC. His work involves using multimodal neuroimaging techniques to explore the system-level mechanisms that govern cognitive function, both in health and disease. His particular interest is in tracking brain activity patterns that reflect communication between regions of the brain. Now, I think of Mac as a young researcher, or that's what it says here. I have this feeling that Mac was here almost as early as myself. As an early PhD student, it's been such a great pleasure to see someone who was working in his early days with Simon Lewis and the group here earlier on, have a really interesting multidisciplinary, interdisciplinary career at that stage, go away, be very successful. We are always very grateful when people of Mac's quality return to continue to contribute to our work. Now working with Glenda Halliday on the neural basis of cognition and dementia. In a big picture way though, and I was reading something that Mac wrote recently, he's trying to go to the big issues. And you know, what other structures and integrative structures might actually be operating within those much larger circuits that Thomas was just describing. So Mac's talk this afternoon is the thalamus. The thalamus integrates the macrosystems of the brain to facilitate complex, adaptive brain network dynamics. Welcome back and welcome again, Mac.
Thank you very much, Ian. It is really a pleasure to be back. As Ian said, I did my PhD at the Brain and Mind Centre and really kind of grew up in many ways there. And then to go away and come back, it's really fun to sort of be on this other end of the spectrum. It's also worth mentioning that I don't think a single person in science ever wants to go after a Nobel laureate. So I'm going to do my absolute best to try to convey some of our recent work. So I'm going to try now to share my screen and let's see if it'll give you, hold on a sec. It seems that it has killed my PowerPoint. All right. All right. Can I get a thumbs up or a nod or something that is now on the screen? Just dreaded silence.
All good.
All good. Excellent. All right. So I'm going to take the bait that Thomas just laid out for early career and emerging early mid-career researchers, which is to be bold. I actually have a background in clinical medicine and made the conscious and some would say unwise decision to leave the solidity of a career in medicine to do a PhD. And I can say without any doubt in my mind that it was absolutely the best decision I ever made. Research is a fundamentally enriching and rewarding career, so long as you, I think, chase after the questions that are the most fascinating to you. And the question for me at the end of the day that really started all this off was trying to work out what are the rules that govern the functioning of the brain. In the same way that we can describe the heart as a pump or the liver as a filter, what kind of language would we use to describe the brain as a system? And it's obviously an incredibly complex question. And all you need to do to think about that is just watch the brain in action. So this is a video of calcium transients from a larval zebrafish. They have the unique characteristic that they are translucent for a number of weeks early in their life. And if you inject the appropriate promoter into their cells, every time a little bit of calcium goes into a cell, which is when the potential raises in the cell, you actually can see that image fluoresce. And so this is just a beautiful video of just some spontaneous activity in the zebrafish's brain. And at once you can see a number of really, I think, striking things. First of all, it's incredibly complex. It's ever-changing and dynamic. It's distributed in a really important way. There's no clear logic like in the heart of a pump that flows back and forth between atria and ventricles. So this incredibly rich activity, yet we have this struggle on the other hand to try to come up with the simple rules that govern the systems level perspective. Maybe one place where I differ with Thomas is that I'm not sure that systems neuroscience has come to its logical conclusion just yet. I think we have a lot yet to say from that systems perspective. So if we think of the challenge that we face as neuroscientists, particularly of the human brain, We could think of what kind of questions we would like to ask of the system. Do we want to look at its function? Do we want to look at its structure? Do we want to look at individual differences? Do we want to look at different diseases with pathology in particular areas of the brain and try to work out which functions those people aren't capable of? So there's many, many different ways that we can study the same system. And I'm absolutely not trying to say that any one of them is any better than the others. But I think in general, as a group of human neuroscientists, we've been a little bit waylaid by our focus on the cortex. So this is a picture from what is actually a really beautiful study from Alex Huth in Nature a few years ago. Essentially what they did was they gave a small number of people, they put them in a scanner, they measured blood flow in their brain while they listened to long stretches of audio. And the idea was that they were trying to capture all the different subtle different contexts and words and ideas that were inherent within that audio. I think it was a podcast. And so what they could then do is once they broke the brain, the podcast up into tiny little bits, they could then regress the activity back against the blood flow within the cerebral cortex, each little vertex within this broader surface. And then they could then assign to each of those vertices a weighting that suggested the particular content that they were listening to at the time. And so then what they can do is they can have this beautiful picture where they map on, fabric over here and then parking over there. And you can go through and search through all your favorite little different terms. Now, this is fine. And this is one way to think about it. But often the conclusion that we come to when we think of these different studies is really that the function somehow lives in the cortex. If I was to sort of grab that little bit of cortex, somehow the ability to perceive a bedroom would be sitting right there in that spot, or, the idea of looking at an apartment would be over here. And I actually think that's a misnomer because the cortex is really not the brain. We often take them for one-to-one, cortex equals brain, but it's easy to forget that the cortex is actually supported and constrained and shaped by a huge amount of subcortical, cerebellar, brainstem structures. They're actually being preserved over a really, really, really long period of evolutionary time. Nowhere is that more clear than in the animal kingdom. So here's a picture of Bambi, and then, oh look, here comes a crocodile jumping out of the water. It's able to imagine, let's say, the Bambi's particular mindset. It can try to predict at the right time. It can understand that it has to camouflage itself, so that it has to come before the deer can see it, and unfortunately it doesn't catch it this time. And if we look at a schematic of the crocodile's brain, the area of cortex is this area in purple. It's really dedicated to olfactory and smell function. And the rest of their brain is, cerebellum, tectum, thalamus, and other parts, very, very small parts of cerebral cortex. So the argument is that actually, maybe by looking at the cortex and forgetting all of the rest of the subcortex, we've essentially sort of we've made a bit of a parts whole mistake. And what we ought to do is maybe step back at that systems level and think about what are some of the other really crucial structures that might be supporting and shaping and constraining the cerebral cortex. Now, the title of my talk essentially betrays this next jump, which is that I want to talk to you about the thalamus. So this is an incredibly important structure in the middle of the brain. It is connected to a huge number of other major structures in the brain. The cortex, it has major connections with the cortex, the striatum of the basal ganglia, the superior colliculus, the inferior colliculus, the hypothalamus, the hippocampus, the cerebellum, the brainstem. This is a structure sitting really like the hub in the spoke of a wheel, sitting right in the middle of the whole thing. So what do we know about the thalamus? Well, we've known for a long time from anatomical studies, but also from human neuroimaging, that the thalamus is really strongly interconnected with the whole cortex, and in a particular way. The connections are essentially mirrored. So you can see the anterior part of the thalamus has strong connections with the frontal lobes, the more posterior part with the parietal lobes. There are smaller nuclei within the thalamus that connect to areas like the occipital and the auditory cortex. Really, there's this spread of connectivity connecting the thalamus with the cortex. But if we go and look a little bit closer at the circuitry of the thalamus and its interactions with the cortex, we actually realize that the connections aren't always one-to-one. So this is some beautiful work by Ted Jones. He literally wrote the book on the thalamus. He has a textbook called The Thalamus that he posted and published before his unfortunate death a number of years ago. But Ted Jones was really enamored by the circuitry of the thalamus and its interactions with the cortex. And what he did was he went through and had a look at essentially tracking individual cells within the thalamus and how they interacted with the cortex. And in general, there's really not two groups per se, but rather a spectrum. And the idea is that the circuits can exist along either one end or the other. So on the left-hand side, we have what Ted Jones called the core thalamus. And that's really what we traditionally think of as the thalamus when we read about it in the textbook. You have a little thalamic cell that's, let's say if it was sitting in the lateral geniculate nucleus, it might receive an input from the retina, that maybe that codes the activity on surround activity from a particular part of the visual field. And what that's going to do is it's going to project up into the cerebral cortex, and it's going to innervate one of these pyramidal cells along with an interneuron. And the idea here is that it's doing something that people would say is akin to message passing. So the idea here is you've got this core thalamic cell, and its two main functions that people like to ascribe to it are message passing and gating. You have to forgive me for the dad joke. So the idea is that this cell is really, really well set up to take an input and to propagate it onto the cortex. The gating function is because it connects to interneurons, which are inhibitory. And the idea is that if it innervates those interneurons, it can inhibit other structures. So it can have a normalizing effect, where it either lets something through or it doesn't let something through. In contrast, there's an entire other population of neurons in the thalamus that have a completely different connectivity pattern. This population was called the matrix thalamus by Ted Jones. And the idea here is that instead of propagating something signal up directly to one particular group of neurons within the cerebral cortex, this group instead projects up and rather diffusely innervates a large number of pyramidal neurons, and in a different layer of the cortex. They typically make contact in the supragranular layers of cortex, which is where all the apical dendrites of these really big pyramidal cells are. And they're often the layers where the feedback processing from higher cortical regions come. So one of the problems with this structure is that we don't really know what its function is, in the same way that we have a really good handle on the idea of this message passing analogy for the core nuclei. The question is, what is this matrix thalamic nucleus doing? It doesn't have the same logical message passing structure. If anything, it looks like it sort of somewhat randomly connects to pyramidal cells. So what function can it have? I went searching for GIFs that look like Bill Gates, and I found one of Steve Ballmer dancing. And it actually ends up being a pretty good analogy for what we think the matrix thalamus does. But maybe I'll try to convince you with something more than just GIFs. What I'm going to try to do is show you a couple recent computational modeling projects that have been conducted by two brilliant postdocs that have been working in my lab. Eli and Brandon are slightly different than a traditional neuroscience postdoc in that they both have backgrounds in statistical physics. So when I started running my own lab, I thought long and hard about what kinds of questions I wanted to answer. And I realized that I could do a lot of neuroimaging experiments. I had a lot of experience with that. I got trained in a really great lab in the US in how to do functional brain imaging experiments in a good way. But what I really wanted to do was to link those patterns to computational models, to basically take the circuits and concepts of circuits inside the brain and to try to understand what the organization of the brain could do when we measured it at that higher level. So what I'm going to do today is walk through very briefly two projects that these two postdocs have been working on. And they both have different vantage points. So Eli's interest is really in treating the brain really from a bird's eye view. And he does something which we call neural mass modeling. So the idea there is that you take a small circuit, here I'm showing it on the left in this little column, which we'll say stands in for a corticothalamic column. So this is going to be like the traditional core corticothalamic column. You're going to have little cortical cells in the top half of the cylinder and thalamic cells in the bottom, each with excitatory and inhibitory populations. And they're going to be wired up in a way that we think respects and corresponds to the typical wiring of this kind of a circuit. Now, this little circuit here has been really extensively validated using electrophysiological markings. And so what we can basically do is take that little circuit, which we think is a pretty good representation of sort of local dynamics within the brain, and then we can wire it up in particular ways that let us then turn little different parameters to basically mimic what we think the matrix thalamic nuclei might be doing. So this is a little bit like the models that Michael Breakspear showed you yesterday in his keynote, but with a little bit more constraint on the kinds of populations that are arising and the interconnections between them. So the really brilliant insight that Eli had when we were talking about this originally was that there's a really direct analogy between the matrix thalamic system and the functions it could have on the cortex and a glass of water. Now, that seems kind of flippant at the start, a glass of water, what could be going on there? But the answer is that there's actually really interesting relationships between the inherent structure of water molecules, how they interact with one another, and the ambient temperature that is applied to the external of the glass. And the idea that Eli had was, as the temperature is raised in a glass of water, what starts to happen is that you get more and more collisions between little water molecules. This is shown on the left-hand side of the graph. But it's not just that you get more collisions, but that there's a certain tail of that distribution that starts to spread out. So there'll be one little water molecule that just accidentally gets bumped into just a few more times in one little window of time. And so that little water molecule will feel like the temperature of the glass is even hotter than it actually is. Because the temperature of the glass isn't actually that hot, it'll quickly realize its mistake and fall back down into the herd. But this was really all we needed, because what we could then do is we could take the logic of this interaction where the physics is really well described, and we could map that onto our model of the brain, very abstract as it is, and we could ask, is the corticothalamic system with these diffuse inputs, the matrix inputs, moving the system in the same way that water is heated up by, let's say, a Bunsen burner? And so, lo and behold, we found pretty much exactly what we were expecting to find in that regard. So the idea was, we swept this particular parameter. This is here, the chi parameter is the proportion of diffuse coupling that we had between the system, which is essentially the stand-in for the matrix thalamic nuclei. And what we're plotting on the y-axis here is the percent of neurons that had crossed their bifurcation. Now, this is a term from dynamical systems. What it's basically telling you is that when at rest, this particular loop, the corticothalamic loop, is essentially enslaved to what we call an attractor. On the left-hand side, you can see that the reticular nucleus is much, much stronger than the sensory relay nucleus. You can imagine that like keeping the lid on the thalamus. And so it doesn't allow the propagation of an emergent rhythm between the S and the E populations in that corticothalamic loop. But as we start to turn up the diffuse coupling, like raising the temperature, what we start to do is we start to allow just a little bit of that population to pop up into the high firing mode, which on the right-hand side, you can now see that the sensory relay nucleus has overcome the reticular nucleus. It can now get its message back to the cortex. And the idea is that as we start to increase diffuse coupling more and more and more, what we're doing is we're sort of opening the lid. We're allowing more and more connections and coalitions to form between neural populations that were otherwise silenced and inhibited. Now, obviously, if we turn this up too high, if we turn the temperature too high, we transition the whole system into a mode that honestly looks a lot like epilepsy, where you've got constant firing and synchrony amongst the population. I won't go into the details here, but Eli also did a number of other really, really cool analyses on that data. That's one of the benefits of doing computational modeling. And just one of the points I wanted to highlight is that right in that, what we call that quasicritical regime, where some of the system has transitioned, but most of it lies back behind, sort of orienting and anchoring it to the kind of subcritical regime. What you can start to see is a separation of timescale. So some neural regions, if they receive an input, really trail back down to zero, and other regions can blow up and have a lot of activity. And so it's almost responsive maybe for the different kinds of sensitivities that we might have to different inputs and different stages of arousal. So that's Eli's work. Brandon's work is focused on a completely different resolution. Now, as we probably gleaned from hearing Thomas's talk before mine, which is really at the systems level, the brain is organized across an astounding number of levels of organization. And Brandon's interest was trying to go, instead of, in the opposite of what Eli did, instead of taking the bird's eye view, what Brandon tried to do is dive right down into the circuitry. And so in this little figure I'm showing here, you can see this matrix thalamic cell in red coming up and making contact with this, this pyramidal cell in orange. And so what Brandon was really interested in doing was taking that model and trying to understand it in detail at that cellular level. And so here on the left, I'm showing you the neuron that we're really interested in modeling. It's called a layer 5 pyramidal neuron. So these are the really big neurons in your cerebral cortex. They're one of the only neurons that spans the entirety of the layers of the cerebral cortex. And they're also really interesting because they project out of the cortex down into the subcortex. So they'll contact areas like the brainstem, the pontine nuclei, which then project into the cerebellum, down into the spinal cord. Another really fascinating thing about these cells, and this is really beautifully described in elegant work by Matt Larkham and his group, is that they have this really interesting feature, which is that the apical dendrites, the areas that receive the feedback inputs in red, and the basal dendrites, the blue regions right around the cell body that receive the feed forward inputs, often from lower areas of cortex, are actually separated out from one another electrically. The idea is that there's this little channel called an HCN leak current, which is open. So that means that any feedback that comes in and hits the apical dendrites while that leak channel is open, any excitability, any of the little ions that get released are just going to leak out into the extracellular fluid, and they're not going to change the way that the system actually responds. In contrast, if you close that HCN channel, which it turns out you can do with things like noradrenaline or acetylcholine, what that's going to do is it's going to make the feedback relevant. It can actually transition that cell into a burst firing mode. Now, all of a sudden, it can push out a lot of outputs, a lot of spikes, and potentially have more of an influence on the rest of the network. And so what Brandon was interested in doing was taking that concept, using what we call Zellovitch neurons, which are a Hodgkin-Huxley model. So like the old idea of looking at every single little ion channel, and they're a reduction on that, which means that they try to work out how that system can act, and they try to simplify it by only measuring one or two really key variables. So we have a bunch of those little cells, and then Brandon basically tweaks two different parameters. So the sigma parameter here is telling us how diffuse or targeted the input is to this apical layer. You could think about this like the matrix thalamus on the right and the core thalamus on the left. And then this probability of bursting, which is going to be the likelihood that this HCN channel is closed and then lets the system transition from regular to burst spiking. And so what Brandon's then able to do is to wire up one of these models and then look at the output at the systems level, right? So we now have a little group of these little neural patterns, and we can move the dials of this different model. And one of the main things to notice at the start is that it's not just sort of blanket similar activity across the whole parameter space. By tweaking these two different parameters in a really, really simple model, Brandon's able to create really interesting heterogeneity in the output. I don't have time to really go into the details, but one of the really interesting findings is that the combination of intermediate bursting over here on the, as we move halfway up this y-axis, and diffuse coupling is able to transition the brain into an integrated state, or the model at least into an integrated state. And that's really consistent with what we've seen before in human neuroimaging. This is from a paper I did back during my postdoc, where we found that brain network integration was really a good measure of cognitive performance. When you were in a task, having to think about variables and track them over time, your brain became interconnected and integrated, suggesting that one of the mechanisms that might drive that is this matrix thalamic or diffuse inputs to the cortex. That's not the only measure that we were able to look at. Brandon was able to calculate a number of other measures, a couple that might be of interest. One of them is the complexity, or the Kolmogorov complexity of the signal. This is a statistical measure that tells you how big of a dictionary you'd need to create in order to recreate the time series. So basically it means that there's more variable and rich activity as we move up the burst probability. And this metric on the right is integrated information, which was really popularized by Giulio Tononi. And the idea is that a brain isn't exactly like a camera, right? A camera takes in a lot of information. It measures activity in the external world and it represents it, let's say, in the activity of a particular pixel, but it doesn't do anything with that information afterwards. And Tononi's insight was that something about the brain's dynamics was that that was really interesting, was that it was doing something with the information. And we essentially found that a measure of that, a quantification of that measure was actually increased in a very similar zone over on the right-hand side of our graph. To validate that more extensively though, Brandon actually grabbed an open data set of a macaque monkey that had its entire left hemisphere covered in what's called electrocorticography grid. So we basically have a measure of the local field potential directly from the cortical surface, while the animal transitions from a sleep state here in blue, a transition waking period in mustard, and then an awake period in red. And then what he could do is take the measures that he calculated from the monkey, and then he could actually port them back onto our brain modeling space. And he essentially found really clear evidence for unique zones that characterize the sleep, the waking, and the awake state. So this is really exciting. It suggests that we can take a really simple model that has been empirically linked to things like conscious awareness and arousal, and we can actually show some of the mechanisms of why it's actually able to give the system, to imbue the system with that capacity. All right, so in other words, the little interim conclusion here, that the matrix thalamus, when we ask what's its function, it's essentially to shape and constrain brain state dynamics. It's really, really well set up not to pass messages, but rather to allow the messages that are passed a chance to get passed around. And that's actually, I think, a really, really important feature of arousal and conscious brain states. Another thing that I really wanted to quickly point out, because I think it's really good to think about when you're thinking at the brain at the systems level, is that the thalamus isn't working on its own. Right? You recall from the start I mentioned that the thalamus has connections with the cortex, but also many other subcortical structures. And two that I just want to highlight here, because I think they have really interesting implications for the way that we think about different diseases that we see in the brain and mind sciences, is that there are actually really strong and conserved connections between the cerebellum, the basal ganglia, and the thalamus, and they distinctly contact these two different populations, but in a way that's maybe slightly counterintuitive. So the first one I'll cover is on the left, the cerebellum. So it's this really important structure, has over half the cells in your brain. It receives input from the cortex via the pontine nuclei. It has this huge overrepresentation of all those inputs, and then it can respond back by feeding its output to the core thalamic nuclei in the ventral tier of the thalamus. Now, a really interesting implication of that is that the cerebellum, we know for a long time, is involved in automaticity and our ability to perform skilled movements. And so one interesting implication is that the cerebellum is now able to support in humans the granular prefrontal cortices that we use for things like decision-making, abstract reasoning, counterfactual reasoning, and things of that nature. And so maybe this circuit here is one of the really key elements that gives us our ability to conduct intelligent conversation. On the other hand, the basal ganglia doesn't contact those cells whatsoever. In fact, it actually preferentially contacts the matrix cells. Now, why is this a bit surprising? Well, for a long time, the sort of general idea of what the basal ganglia does is that it gates loops. It allows the thalamocortical loop to oscillate. So basically, the function of the basal ganglia has been mapped onto the core circuitry, not onto the matrix circuitry. So the question then becomes, well, what could this function be? And it's actually, I think, quite fascinating. What the basal ganglia can now do is it can release a little bit more of the matrix thalamic nuclei in a particular location, such that that allows that little area to transition to become the one that now has the influence over your behavior. The idea is that it can help you to swerve out of what you were going to do, let's say your inbuilt automatic response, the thing you'd learned before via the cerebellar circuitry, and it allows you to move into a more deliberate mode of processing. Now, this is really fascinating. It's really early days. But it turns out that mechanistic models of free will and agency really involve something just like this. The idea of using the constraints of the system to inject just a little bit of randomness. Not too much, because then you do absolutely anything, but enough that you're doing something that is constrained by your previous experience. And I'm absolutely fascinated by the idea that these kinds of capacities might be driven in part through the corticothalamic circuit as it's mediated by the subcortical circuitry. In case you don't want to take my word for it, there's really beautiful work on birdsong that basically really conveys a lot of these really crucial take-home messages about subcortical influence over complex behaviors. The one that I wanted to leave you with was that the basal ganglia in birdsong is actually important in two very critical phases. One is early in the course of the bird's life when they're learning their father's song, and they basically have to have the basal ganglia in order to be able to have the grist for the mill in order to learn how to recreate that song accurately. But it turns out that later on in life, it's actually not important for that reason at all. What it's important for is actually improvising on the circuit that already knows and creating its own song. And so in a way, the basal ganglia has this really important role to play, but it might be different at different stages of learning. I think this might help us, give us a language for how to describe some of those influences. All right, so in summary, what I've really tried to convey today is that taking this systems level perspective of the brain and really thinking about subcortical structures and their influence on the cerebral cortex can give us a real insight into how some of the emergent dynamics might arise. It's not that the cortex is sitting on its own, like in a sort of Spockian sense, divorced from the world, doing logical reasoning, but rather that activity and learning shape and constrain these circuits so that they're brought to bear at the appropriate point in time. And I think, at least for me, the thalamus plays a really crucial role in constraining those inputs and shaping which of them will actually drive behavior. And so I'll bring you back to this beautiful picture of the zebrafish brain spiking away, bursting, moving. if we can think about how the circuitry of the brain and the kinds of constraints that are placed on it allow this to perform its different functions, it's a little bit like getting a new vantage point on the brain. This is some beautiful footage of a series of shots that were taken, and then each of the different points of the Milky Way were then mapped together, such that the Milky Way stays put and the Earth moves. And I shouldn't have to remind you that that's exactly what reality is, but to us, it looks like the sky moves overhead. So sometimes looking at things in a different way can help to understand the true nature of the system. And with that, I'd like to say an extra large thank you to Eli Muller and Brandon Munn, who did the bulk of the work on these studies today, as well as the rest of my collaborators.
Thank you so much, Mac. And it is not easy to come into bat, after somebody's won a Nobel Prize, but I think in a very Glenn Maxwell-like way, you did a marvellous job of that.